Hi, my name is John Poole. I founded our population genomics lab at the University of Wisconsin almost 10 years ago. And in this video, you'll get to hear a little about the research we have going on. So in the big picture, our lab uses genetics and genomics to ask fundamental evolutionary questions. The questions that we're most interested in range from what adaptive evolution looks like at the genetic level to the very earliest stages of reproductive isolation to the relationship between genetic diversity and adaptive potential. Often in this work, we use Drosophila as a very powerful model system that allows us to make faster progress in this research than probably any other organism. And we take a range of approaches. We do collect and analyze large next generation sequencing data sets for population genomic studies, for quantitative genetic mapping, for RNA sequencing, and so on. Of course, we spend plenty of time analyzing that data, but we also perform purely computational studies, often featuring large-scale population genetic stimulation, and sometimes developing our own new methods for analyzing genomic data. So our lab resources include a fly room with thousands of Drosophila stocks, along with a molecular lab. We also have shared lab computers. Plus, we're often running thousands of jobs at any given time on the university's computer network. In the lab, aside from myself, we currently have one postdoctoral researcher, four continuing PhD students, and four undergrads. We've also just had two PhD students defend. Plus, we've had five former postdocs in the lab, along with many undergraduate alumni. And now I'll give you a chance to hear from some of our current members.
Hi, my name is Quentin Springelmeyer. I'm actually recently defended my PhD dissertation, and for that research, I did a couple of projects. The first being I looked at the population history of Drosophila melanogaster and how they expanded within and out of Africa. And this study provided a nice demographic model of where this fly originated in sub-Saharan Africa, and then how it expanded up throughout Africa and then over into the Middle East and out into Europe. My other projects I worked on was looking at the genetic basis of adaptive traits. One was ethanol tolerance found in two cooler high altitude populations, Ethiopia and South Africa, and then a higher latitude population of France. And then I also looked at the genetic basis of body size in the population of Ethiopian highland flies. And these flies have the largest thorax and wing size of any known populations. And from these two studies, we were able to perform QTL mapping and find that these traits are actually moderately polygenic, and with the hope that now further research could look at exploring these candidate genes found within these QTL regions and maybe provide functional testing.
Hi, my name is Thiago. I'm a PhD student. I'm studying the genetic base of adaptation. One part of my project is trying to understand the power of SNP-FST to detect loci under selection, in contrast to other approaches that use the whole window FST. And this might be very useful to tell us about different kinds of selection that are happening in the population, different soft or hard sweeps, complete or incomplete sweeps. And another side of my work is also dealing with adaptation genetics, but now I'm looking at the genetic architecture of adaptive traits. So using pigmentation in Drosophila as our model, we're trying to understand how many different QTLs, how many different loci are underlying this trait in a population that has been selected to become dark. And have a lot of evidence already that different flies from the same population will have different loci underlying this trait. So we're trying to see how polymorphic these QTLs are and how they interact with each other. Also trying to understand a little bit about the epistasis among these traits. So this is my general project here in the Pool Lab.
Hi there, this is Suyan. I'm doing my first year of PhD and I'm currently studying the genetic basis underlying local adaptation of Drosophila suzuki along its invasion routes. So these evoluted creatures are an attractive biological model to study invasion process, and it's also highly destructive pest in fruit production, which caused 1 billion loss per year. During the last decade, Drosophila suzuki has spread from Eastern Eurasia to nearly all temperature and subtropical regions of the world, as well as parts of the tropics. Given such invasion success, we're really curious about how they've adapted to different new environments and how they've obtained resistance to insecticide. And we're going to use whole genome sequencing to answer these questions by looking at associations between genotype and environment, and perhaps also variations and insecticide resistance genes.
Hi, I'm Chris McAllister. I'm a grad student in the lab. I work with the lab's wild Drosophila melanogaster from Zambia in Africa, which have many stretches of inverted DNA similar to the inversion depicted on the left. These inversions aren't common in high altitude Ethiopia or temperate Europe. Because these inversions have maintained intermediate frequency in lowland populations despite bottlenecks in migration, we hypothesize that the inversions are maintained by some form of balancing selection. Specifically, I'm testing whether alternate inversion orientations trade off mating display and survival phenotypes. These could be balanced when different alleles of loci in the inversions trade off between the benefits to mating displays, pictured below, where the benefits are frequency dependent, and benefits to survival, which are independent. To study this, I've written population simulations that simulate mate choice explicitly, which confirm the possibility of this dynamic. I'm following this by tracking the inversion frequency within an outbred population between parents, embryos, and adult offspring to check for differential fitness effects across reproductive competition and demographic stages. I hope to demonstrate this kind of balancing selection is surprisingly common, where among alleles with pleiotropic trade-offs, the combined fitness of multiple alleles scales differently between the phenotypes affected. The scaling disfavors intermediate haplotypes and causes selection for linkage in inversions. The example systems here are all populations with differentiated inversions that underlie phenotypic differences with fitness trade-offs that I think might be explained by this pleiotropic dynamic.
Hi, my name is Matthew. I am a graduate student in the lab. My project is concerned with discovering the genetic and biological underpinnings that define the earliest stages of reproductive isolation. Species are often defined by their similarity and ability to produce fertile and viable offspring. Reproductive isolation is the inability to produce such viable or fertile offspring. Individuals within the same species can become reproductively isolated from one another when there is an event that restricts gene flow between groups within a population. One such event may be the occupation of a population in different geographic locations. If this geographic distance is sufficiently far or cut off from the ancestral population, gene flow between these groups may become very limited or even completely restricted. Over time, this allows for forces such as mutation, adaptation, and drift to alter the genetic architecture of these populations. Populations may become sufficiently differentiated such that upon rehybridization, individuals from one population may no longer be able to produce viable offspring with individuals from one another. Using Drosophila melanogaster as a model organism, we aim to describe this process in its earliest stages. We use populations of fruit flies from the ancestral range of Zambia and a more recent population from France to generate population hybrids. These groups have split from one another only very recently on an evolutionary timescale. We studied the genetic basis of hybrid male sterility using unique mapping approaches to identify potential Dobzhansky-Mueller incompatibilities. Additionally, we use multiple behavioral assays to study the biological basis of this sterility. Among these include courtship behavior, neurological functionality, and the assessment of sperm quality and proper transfer to females. These approaches allow us to begin to answer longstanding questions about the forces that govern speciation during its emergence.
Hi, I'm Maud Duranton. I'm working as John's lab as a postdoc, and I'm working on the relationship between integration and recombination. When two populations diverge over time, they can sometimes exchange genetic material if reproductive isolation is not complete. In many species, it has been demonstrated that the amount of exchange genes tend to be higher in genomic regions where recombination is high. This positive relationship is due to the effect of purifying selection that can easily get rid of deleterious material in highly recombining regions. However, between the European and African lineages of Drosophila melanogaster, we observed the opposite patterns. To try to understand what creates this correlation, I'm using whole genome simulation. In my simulation, I have two populations, P1 and P2, that contribute in a certain proportion to form a third population, P3. These two populations contain a certain amount of positive mutation, here 1,000, with a certain selection coefficient. After 1600 generations of reproduction, I look at the proportion of genetic material coming from P2 within the P3 population, depending on the proportion of mutation present in each population. And I showed that under certain conditions, there is a negative relationship, indicating that positive selection might be involved between the European and African lineage of Drosophila melanogaster.
And hello, this is John again. For my own part, mostly I do typical professor things like meeting with my lab members, writing grants, teaching, and committee service. But I have managed to do some research of my own here, as you can see from our list of publications. And I'm working on getting a new version of my bulk segregant QTL mapping software out soon. If you'd like to learn more about any aspect of our lab, feel free to get in touch with me. My email is jpool@wisc.edu, and I'd be glad to hear from you.